Night blindness is a condition that makes seeing in low light difficult or impossible. It's often a symptom of other eye diseases, and it can be dangerous in many situations, such as this one. Pretty scary, right? Okay, that's better. Researchers are trying to better understand the mechanisms of night blindness so that we can identify targets for therapeutic treatment. Let's take a look at the first step of vision processing, how the eye converts light into a nervous signal. This is called transduction, and it takes place in the photoreceptors. Rhodopsin, a G protein-coupled receptor found in the photoreceptors of the eye, is a key player in the visual system because it triggers activity in response to light. It goes through a cycle of decomposing and reconstituting in response to light. So, without light, its conjugated form consists of 11 cis-retinol and the protein scotopsin. When it's exposed to light, the cis-retinol gets converted to trans-retinol, and the rhodopsin breaks down into parts, including meta-rhodopsin 2. This activates transducin, which activates phosphodiesterase, which converts cyclic GMP to GMP, so that the sodium channel closes and the cell hyperpolarizes. Okay, so how does rhodopsin get reconstituted and maintain its sensitivity to light? This cascade gets turned off when GRK1, or G protein receptor kinase 1, phosphorylates and deactivates meta-rhodopsin, and arrestin-1 binds to the receptor. This promotes the conversion of trans to cis-retinol so that rhodopsin gets regenerated. So how is all of this related to night blindness? Well, it's often an issue if you're lacking rhodopsin because it serves as a light receptor. Animals better adapted to the dark generally have more rhodopsin, and this is often generated from vitamin A. Vitamin A is also known as transretinol, which is convertible to transretinal, which can then be converted to cis-retinol in the regeneration of rhodopsin. So people who lack vitamin A can suffer from night blindness because they can't generate enough rhodopsin. What are some other things that could affect the regeneration of rhodopsin? Another part of this picture that's being looked at is the GRK1 phosphorylation and arrestin binding, since this promotes the conversion of transretinol to cis and the regeneration of inactive rhodopsin. A recent study looked at two rhodopsin mutants known to cause night blindness in humans, G90D and T94I. So here we're looking at the active site of rhodopsin. One mutant is G90D, meaning at position 90 you have glycine, or G, replaced by aspartic acid, D. Same deal for the T94I mutant. So at the 94th amino acid, threonines replaced with isoleucine. Both of these cause night blindness by constitutively or constantly activating transducin, which keeps the phototransduction cascade turned on so that the photoreceptors are desensitized to light. They found that both of these rhodopsin mutants are effectively phosphorylated by GRK1 and its subtypes, so this part of turning off the cascade seems to work okay. But the G90D mutant demonstrated significantly lower arrestin-1 binding, while the T94I mutant bound arrestin normally. So from this, the researchers hypothesized that the reduced binding of arrestin-1 to the G90D mutant is one of the mechanisms that contributes to night blindness in humans. But the fact that the T94I mutant bound arrestin normally suggests that night blindness is brought about by more than one mechanism. It makes sense to look at the regeneration of rhodopsin as a pathway through which issues could cause night blindness. But this picture is pretty complicated, and there are many ways rhodopsin activity can be altered. There are also several diseases that cause night blindness, and we still haven't figured out how to treat many of them. But at least we can get an idea of the mechanisms of night blindness at the cell level.